On a May morning in 1796, in the English county of Gloucestershire, a country physician took material from a blister on the hand of a milkmaid named Sarah Nelms and scratched it into the arm of an 8-year-old boy named James Phipps. The material came from a cowpox pustule. The physician believed, based on years of careful observation and a specific local tradition among dairy workers, that people who had caught cowpox from their cattle never subsequently caught smallpox. If that was true, then deliberately infecting James Phipps with cowpox might protect him from smallpox, the most feared disease in the world, the disease that had killed an estimated 300 million people in the 20th century alone, the disease whose characteristic pustules had scarred the faces of pharaohs and emperors and ordinary people alike across 30 centuries of recorded human history. The physician was Edward Jenner. The procedure he performed that morning was the first vaccination in recorded history. The word vaccination itself comes from the Latin vacca, cow, in honor of the bovine origin of Jenner's cowpox material. And what Jenner did on that May morning in 1796 with a milkmaid's blister and an 8-year-old boy's arm was the beginning of the most consequential medical intervention in the history of the human species. The technology that would eventually eliminate smallpox from the earth entirely. That would reduce the childhood mortality of measles and polio and diphtheria and whooping cough and dozens of other diseases from the catastrophic levels of pre-vaccine history to fractions of their former selves, and that would save, by the most conservative estimates, an estimated 3 to 5 million lives every single year in the 21st century. The story of vaccination does not begin with Jenner. It begins thousands of years earlier, in the specific human observation that surviving a disease once appeared to protect against catching it again, an observation so ancient and so universal that it appears in virtually every medical tradition on earth. The ancient Greeks noticed it during the plague of Athens in 430 BCE. Thucydides recorded that the survivors of the plague were the only ones willing to nurse the newly sick, because they had learned from experience that they would not be struck a second time. The ancient Chinese and Indian and Persian medical traditions had all made similar observations, and some had gone further, had developed specific techniques for deliberately inducing mild versions of dangerous diseases in the hope of producing protection without the full lethal consequences of the natural infection. The most developed of these pre-modern immunization traditions was the practice of variolation, the deliberate inoculation with material taken from mild smallpox cases to induce a hopefully mild form of the disease that would protect against the more severe natural infection. The practice was widespread in China by the 10th century CE, where dried smallpox scabs were powdered and blown up the nostrils, or material from mild cases was scratched into cuts in the skin, and had spread through the Ottoman Empire to the Middle East and North Africa by the 17th century. It reached Europe through the specific diplomatic channel of Lady Mary Wortley Montagu, the wife of the British ambassador to the Ottoman Empire, who observed the practice of variolation in Constantinople in 1717, had her own children variolated, and campaigned on her return to England for the adoption of the practice in British medicine. The Royal Society examined the evidence. The Princess of Wales had her children variolated. The practice spread among the European aristocracy and eventually the general population, imperfect, dangerous by modern standards, causing actual smallpox deaths in a small but real proportion of those inoculated, but reducing overall mortality from the disease significantly in the populations where it was adopted. Jenner was born on May 17, 1749, in Berkeley, Gloucestershire, the son of a Church of England vicar, the eighth of 9 children, orphaned at 5 and raised by his eldest brother. He trained as a physician under the great London surgeon John Hunter, whose specific approach to medicine, systematic observation, careful recording, and the willingness to test hypotheses through deliberate experiment rather than merely accepting received wisdom, shaped Jenner's intellectual formation in ways that proved decisive for his greatest discovery. He returned to Berkeley and established a country medical practice that he maintained for the rest of his life, the specific quiet productivity of a provincial physician who was simultaneously conducting the systematic scientific observations that led to his greatest discovery. His Berkeley countryside gave him the specific local knowledge that London researchers lacked. The dairy workers of Gloucestershire had long maintained that catching cowpox protected against smallpox, a tradition that Jenner heard and took seriously where a more conventionally minded physician might have dismissed it as folk superstition. He spent approximately 20 years investigating the cowpox protection tradition before he was confident enough to test it experimentally. He collected case histories. He noted the specific circumstances under which dairy workers caught cowpox and the specific extent to which they subsequently avoided smallpox during epidemics. He identified 2 different skin conditions that dairy workers called cowpox, of which only one produced smallpox protection. He was careful, more careful than the dramatic simplicity of the May 1796 experiment suggests. James Phipps developed a mild cowpox infection following the scratching of Sarah Nelms's material into his arm, a slight fever and a pustule at the inoculation site, exactly what Jenner had predicted. 6 weeks later, Jenner inoculated Phipps with actual smallpox material, the standard variolation procedure, and waited to see if the cowpox protection was real. Phipps did not develop smallpox. He was immune. Jenner inoculated him with smallpox material again, several months later. Again, no disease. The protection was genuine. He published his findings in 1798, a thin volume at his own expense, having been unable to persuade the Royal Society to publish it in their journal, in one of the more embarrassing institutional judgments in the history of science. The publication was titled An Inquiry into the Causes and Effects of the Varioli Vaccini, the word vaccini from the Latin for cattle, and described 23 cases of vaccination with cowpox and subsequent testing with smallpox material, demonstrating the protective effect with the specific systematic evidence that distinguished Jenner's approach from the folklore it extended. The reception was mixed and eventually enthusiastic. Some physicians repeated his experiments and confirmed his results. Others were skeptical. The mechanism was entirely unknown, the procedure was novel, and the specific horror of deliberately infecting someone with material from a diseased cow offended both the medical conservatism and the religious sensibility of some of Jenner's contemporaries. Satirical cartoons appeared depicting vaccinated people sprouting cow parts. Clergy objected that interfering with divine providence through inoculation was ungodly. None of it stopped the spread. Within a decade of his 1798 publication, vaccination against smallpox was being practiced across Europe and North America. Napoleon Bonaparte, who was conducting his military campaigns across Europe in the first decade of the 19th century, was sufficiently impressed by the evidence for vaccination to have his entire army vaccinated and to name Jenner one of the greatest men in history. The specific irony of Britain's greatest military enemy becoming vaccination's most powerful advocate during the Napoleonic Wars was not lost on Jenner's contemporaries. The mechanism of what Jenner had observed, the specific biological process by which the immune system learned from cowpox exposure to recognize and defeat smallpox, was completely unknown to Jenner and would remain unknown for most of the 19th century. Jenner was a brilliant empirical observer and a careful clinical experimenter, but he was working entirely without the germ theory of disease, without any concept of the immune system as a coherent biological apparatus, and without any understanding of the relationship between viruses and the diseases they caused. He knew vaccination worked. He did not know why. The why, the specific understanding of the immune system and its response to pathogens, required the work of Louis Pasteur and his successors. Pasteur's development of the germ theory of disease in the 1850s and 1860s established that specific microorganisms caused specific diseases, the conceptual foundation without which rational vaccine development was impossible. His development of laboratory techniques for growing and studying pathogenic microorganisms in pure culture provided the experimental tools that vaccine research required. Pasteur's specific contribution to vaccine development was the discovery of attenuation, the process by which a pathogen could be weakened, by passage through a different host or by exposure to heat or other stresses, to produce a form that retained its ability to stimulate the immune response without retaining its full ability to cause disease. The attenuated pathogen could then be used as a vaccine, producing immunity without the disease. His chicken cholera vaccine of 1879, discovered, like penicillin, by an accidental observation, was the first demonstration of attenuation. He had accidentally allowed a culture of the chicken cholera bacterium to age and weaken before injecting it into chickens, and found that the chickens survived the normally lethal bacterium and were subsequently immune to fresh culture. The aging process had attenuated the bacterium, weakened it, without destroying its immunogenic properties. His anthrax vaccine of 1881, Tested in the most famous public demonstration in the history of vaccine science, the Paola-Lefort trial, was the specific moment when vaccine development became a public event of extraordinary drama. Pasteur publicly challenged the skeptics who doubted his anthrax vaccine, proposing a controlled trial in which half a group of sheep and cattle would receive his vaccine and half would serve as unvaccinated controls, with all animals subsequently receiving a lethal dose of anthrax. The trial was conducted in May 1881 before a large audience of scientists, physicians, farmers, and journalists. The vaccinated animals survived. The unvaccinated animals died. The specific theatrical quality of the demonstration, the dead unvaccinated animals laid out in the field beside the healthy vaccinated ones, made it one of the most powerful scientific demonstrations of the 19th century. His rabies vaccine of 1885 was the specific application that made him a public hero of the first magnitude. Rabies, the viral disease transmitted by the bite of an infected animal, causing the specific terrifying constellation of symptoms that includes hydrophobia, agitation, delirium, and inevitable death, had been universally fatal in recorded human experience. There was no treatment. There was no prevention beyond avoiding the bite of infected animals. Pasteur's development of an attenuated rabies vaccine, produced by drying infected rabbit spinal cord material, was the scientific achievement. The specific human drama that made it famous was its first use in a human patient. On July 6, 1885, a 9-year-old boy named Joseph Meister was brought to Pasteur's laboratory by his mother. He had been bitten 14 times by a rabid dog 2 days earlier. The bites were so severe that the physician who examined him initially was certain they would prove fatal. Pasteur, who was not a physician and had never used his vaccine in a human patient, faced the specific ethical anguish of deciding whether to give an untested vaccine to a dying child. He decided to proceed. Joseph Meister received 13 injections of progressively stronger rabies vaccine preparations over 11 days. He did not develop rabies. He recovered completely. He survived. News of the treatment spread around the world. Within a year, Pasteur's laboratory was receiving patients from Russia, America, and across Europe. People who had been bitten by rabid animals and who had traveled to Paris in the desperate hope that the famous French scientist's vaccine could save them. Most of them were saved. The Pasteur Institute, founded in 1888 with funds raised by public subscription, reflecting the specific public gratitude for a scientist who had given the world something genuinely miraculous, became the center of vaccine research that it remains today. The 20th century was the golden age of vaccine development. The specific combination of the germ theory foundation, the techniques of attenuation and inactivation established by Pasteur and his successors, and the specific public health infrastructure that allowed systematic vaccination programs to reach entire populations produced a sequence of vaccine successes that reduced the burden of infectious disease on human populations by an order of magnitude previously unimaginable. The diphtheria antitoxin developed in the 1890s by Emil von Behring, using a different mechanism than attenuated vaccines, employing antibodies produced in horse blood to neutralize the diphtheria toxin, was the first specific immunological treatment for a bacterial disease and won the first Nobel Prize in Physiology or Medicine in 1901. The tetanus antitoxin that followed from the same research program was the specific biological product that transformed the mortality of wound infections in the First World War. The pertussis vaccine, protecting against whooping cough, the bacterial disease that killed thousands of children every year in the pre-vaccine era, was developed in the 1920s. The diphtheria pertussis tetanus combined vaccine developed in the 1940s was the specific immunological package that began the systematic protection of children against 3 major killers simultaneously. The polio vaccine is the story within the story, the specific narrative of vaccine development that has generated the most popular drama and the most public emotional investment of any single vaccine campaign in history. Poliomyelitis, the viral disease that invaded the nervous system and could leave its survivors paralyzed, that killed thousands and paralyzed tens of thousands every year in epidemic years, that was specifically feared by the middle-class American families whose children played in the public swimming pools and summer camps where transmission was particularly likely, was the disease that the American public most urgently wanted defeated in the mid-20th century. Jonas Salk developed the inactivated poliovirus vaccine, killing the poliovirus with formaldehyde and using the dead virus to stimulate immunity, and tested it in the largest clinical trial in the history of vaccine science in 1954. More than 1 million children participated in the trial, the polio pioneers, as they were called, whose parents enrolled them knowing that some would receive the vaccine and some would receive a placebo, and that the distinction would not be revealed until the trial was complete. The announcement on April 12, 1955, the 10th anniversary of Franklin Roosevelt's death, the most famous American polio patient, that the Salk vaccine was safe and effective produced one of the largest public celebrations in post-war American history. Church bells rang. Strangers embraced in the streets. Schools closed for spontaneous celebration. When asked who owned the patent on the polio vaccine, Salk gave the answer that has been quoted ever since as the model of vaccine altruism. Could you patent the sun? There was no patent. The vaccine was freely available to every manufacturer who could produce it. Albert Sabin's subsequent development of the oral poliovirus vaccine, a live attenuated vaccine that could be administered on a sugar cube, requiring no needle, no medical professional, and no sterile injection equipment, was the specific technological form of the vaccine that made global polio eradication campaigns practical. The specific visual of a drop of pink liquid on a sugar cube being given to a child is one of the most iconic images of 20th century public health. The global effort to eradicate polio, modeled on the successful smallpox eradication campaign, has reduced polio cases from an estimated 350,000 per year in 1988 to fewer than 100 in 2021. The smallpox eradication campaign, the specific global effort coordinated by the World Health Organization from 1967 to 1980 that succeeded in eliminating smallpox from the human population entirely, remains the greatest achievement in the history of public health. The disease that had killed 300 million people in the 20th century alone, that had killed pharaohs and Aztec emperors and European royalty and peasant children indiscriminately, was declared eradicated on May 8, 1980. The last naturally occurring case of smallpox was Ali Maumalin, a Somali hospital cook who recovered in 1977. The eradication campaign worked because of the specific properties of the smallpox virus. It only infected humans, it had no animal reservoir, it produced a characteristic rash that made cases identifiable, and the vaccine was stable enough to be stored and transported to remote regions. It also worked because of the specific human dedication of the thousands of public health workers who carried vaccines into the most remote regions of Africa and Asia and South America, finding and vaccinating every contact of every case in the specific containment strategy that ultimately cornered the virus. The development of recombinant DNA technology in the 1970s and 1980s, the specific biotechnology that allowed the genes of pathogens to be inserted into other organisms and the proteins they encoded to be produced in quantity, transformed vaccine manufacturing in ways that Jenner and Pasteur could not have imagined. The hepatitis B vaccine licensed in 1986 was the first vaccine produced using recombinant DNA technology. The hepatitis B surface antigen produced in yeast rather than harvested from infected human plasma. The human papillomavirus vaccine licensed in 2006, protecting against the virus responsible for most cervical cancers, was the specific vaccine that demonstrated that vaccination could prevent not just infectious diseases but the specific cancers caused by infectious agents. And then COVID-19. The pandemic that began in 2019 produced the most rapid vaccine development in the history of immunology, mRNA vaccines against SARS-CoV-2 moving from viral sequence to emergency use authorization in less than a year, a timeline that compressed what had previously been a decade of development into months. The specific technology, messenger RNA encoding the spike protein of the virus, delivered in lipid nanoparticles, instructing the body's own cells to produce the antigenic protein and stimulate immune response, was not invented for COVID-19. The mRNA vaccine technology had been in development for decades, its foundational science conducted by Cottaline Carico and Drew Weissman at the University of Pennsylvania in the early 2000s. Their specific breakthrough in modifying mRNA to avoid triggering the immune reaction that had previously made therapeutic mRNA unstable being the specific technical advance that made the COVID-19 mRNA vaccines possible. The story that began with a milkmaid's blister and an 8-year-old boy's arm in 1796 has produced one of the most consequential sequences of scientific and medical advances in human history. Smallpox eradicated. Polio on the verge of eradication. Measles, diphtheria, whooping cough, tetanus, hepatitis B, human papillomavirus, meningitis, pneumonia. Each of these diseases, which had killed hundreds of millions across human history, now preventable by vaccines that are among the most cost-effective health interventions ever devised. 3 to 5 million lives saved every single year. Every year. By vaccines. By the specific biological technology that Edward Jenner discovered in a Gloucestershire garden in 1796, and that every subsequent generation of scientists has refined and extended and applied to new targets. James Phipps, the 8-year-old boy who received the first vaccination, lived to be 65 years old. Jenner had built a cottage for him and his family, rent-free for life, in the Berkeley garden where the vaccination had taken place. He attended Jenner's funeral in 1823 and is buried near his benefactor in the Berkeley churchyard. He was the first vaccinated person in history. He did not know what had been done to him or what it would eventually mean for the human species. He was 8 years old. He had a slightly sore arm for a few days. And 225 years later, the technology that began with his slightly sore arm has saved more human lives than any other discovery in the history of medicine.